Alright folks, we are on 3-8, which is the cell division cycle, mitosis, and DNA replication. So before we play this video for you, let me just tell you the cell division cycle is exactly that, a cyclic event, a reoccurring event. When I say cyclic, what I mean is reoccurring. For some cells anyway. Some cells leave the cell division cycle and no longer divide, but the cells that are active dividers, they go through this growth phase, and we call some of this growth phase here interphase. And then they go through this mitosis or mitotic phase right here, which is really divvying up the DNA equally. So we call it the nuclear division. You're just making sure that both daughter cells get identical copies of DNA. And then the cell may go through the cytokinesis phase where you divvy up the cytoplasm and hopefully equally. So this is the cell division cycle. So we're going to talk about the interphase, G1, S, and G2. We're going to talk about the subphases of mitosis. You probably have heard of them before: prophase, metaphase, anaphase, and telophase. And then we'll talk about cytokinesis. So here we go.
Most eukaryotic cells follow a process of growth and division called the cell cycle. These events include a growth stage, mitosis or nuclear division, and cytokinesis or division of the cytoplasm. Throughout interphase, the cell is engaged in growth and metabolic activities. Interphase can be further broken down into three discrete phases called G1, S, and G2. During the G1 or first growth phase, normal cell functions occur as well as cell growth. In the S phase, DNA replicates, producing 2 copies of each chromosome. During the G2 phase, the cell continues to prepare for mitosis and cell division. Mitosis, or the M phase, has 4 stages: prophase, metaphase, anaphase, and telophase. During prophase, the chromosomes become visible and condense, becoming shorter and thicker. Each identical copy of a single chromosome is called a sister chromatid. The nuclear envelope breaks down and spindle fibers form as microtubules grow out of the centrioles that move to opposite poles of the cell. During metaphase, the double-stranded chromosomes line up along the equator of the cell. The spindle is now fully formed and the microtubules attach to each sister chromatid. Anaphase begins when the sister chromatids of each chromosome begin to separate. The centromere that holds sister chromatids together divides, and the chromosomes move away from each other along its spindle fiber. In telophase, the 2 groups of chromosomes reach the opposite ends of the cell. As a new nuclear envelope starts to form around each group, the chromosomes uncoil and the spindle disappears. The division of the cytoplasm and organelles is called cytokinesis, or the C phase. The result of mitosis and cytokinesis is the formation of 2 genetically identical cells. To summarize the entire cycle, G1, S, and G2 phases occur during interphase while the cell is growing and preparing to divide. Mitosis, or the division of the nucleus, consists of 4 phases: prophase, metaphase, anaphase, and telophase, and in these phases duplicate copies of the chromosomes are split equally. Finally, the cell's cytoplasm divides during cytokinesis and results in the formation of 2 genetically identical daughter cells.
Okay, that's the cell division cycle in a nutshell. Now, during S phase of interphase, I guess I should be clear about that because I don't think he was. This entire gray area is called interphase, not just G1. G1 is a subphase of interphase, S is a subphase of interphase, and G2 is a subphase of interphase. So interphase is this entire gray area. During S phase of interphase, that's when you copy your DNA. S stands for synthesis. So why we call it S phase is because we're doing synthesis, and we're actually doing DNA synthesis. We're replicating DNA. That's how we get our copies of our chromosomes. We replicate the DNA. And then mitosis is just divvying them up equally. So during S phase, we do DNA synthesis. We call DNA synthesis quite often DNA replication. Now, sometimes we actually call it DNA synthesis, but DNA replication and DNA synthesis are the same thing. Let's take a look at DNA replication.
Prior to cell division, the genetic information which is stored in chromosomes must be copied in a process called DNA replication, and the duplicated DNA separated evenly between two cells. Understanding of DNA replication comes largely from studies of E. coli, bacteria that are found by the billions in the large intestine. Let's take a look at how DNA replication occurs in an E. coli cell. As we zoom in, we see the DNA. At the origin of replication, the two strands of DNA separate and then serve as templates for making new strands. The result is a replication bubble. The bubble grows in both directions, forming two replication forks. In humans, numerous replication bubbles are formed. Let's zoom in on one of them. Some of the proteins that work together at the replication fork are shown here. Here, DNA helicase unwinds the two strands, and DNA polymerases type 3 build new strands of DNA. A critical component of the DNA polymerase III protein is the DNA clamp protein, which prevents the polymerase from dissociating from the template strand. Thus, the process of DNA replication is called semi-conservative. Each new double helix contains one original DNA strand and one new DNA strand. Because strands in a DNA double helix run in opposite directions, the new strands must be made in different ways. One new strand, the leading strand, is built continuously and forms from its 5' end to its 3' end. The orientation of the other new strand, the lagging strand, is opposite to the working orientation of DNA polymerase III, and thus the lagging strand must be built in fragments, called Okazaki fragments, in the 5' to 3' direction using multiple primers. First, let's focus on the leading strand. DNA polymerase III builds a new strand of DNA by adding DNA nucleotides one at a time. Each new nucleotide must pair up with its complementary nucleotide on the parental strand. Adding new nucleotides works the same way on both the leading and lagging strands. Because of the opposite orientation, replication of the lagging strand is more complicated. To initiate synthesis, an enzyme called primase reads the DNA strand and produces a short segment of RNA which serves as a primer. A DNA clamp protein surrounds the DNA and attaches to DNA polymerase III, which builds the rest of the new fragment of DNA until it reaches the previously synthesized Okazaki fragment. When the fragment is finished, the polymerase releases from the Okazaki fragment. How are Okazaki fragments of the lagging strand joined together? A different DNA polymerase I, removes RNA and replaces it with DNA. However, it cannot finish connecting the Okazaki fragments. An enzyme called DNA ligase joins the Okazaki fragments together. Growth of the leading and lagging strands continues at both replication forks until there are two identical, double-stranded DNA molecules. Although bacteria are very different from humans, the process of DNA replication in bacteria is similar to what happens in your own cells.
Okay, that's DNA replication. So let me talk through a couple of things she said. First of all, you have to unwind the original parent strand. It's a double helix. Helicase does that. There's an enzyme right down in here called helicase, which unwinds the double-stranded DNA. And then what we do is we copy each one of these single strands. Now, the parent strand was A to T and G to C, and now these two daughter strands are going to be A to T and G to C. So this daughter strand and that daughter strand, when they're done, are going to be equivalent to each other. They are going to be the sister chromatids, because we always bind A to T and G to C. So you got to remember that the nucleotide bases are A to T and G to C. And a lot of times you'll see them written like this. Why the heck would that be? Well, as it turns out, adenine has a double hydrogen bond, 2 hydrogen bonds with thymine, and guanine has 3 hydrogen bonds with cytosine. That's why they write it like that. You can see the nucleotides come in one at a time and are in hydrogen bond and are covalently attached until you get your 2 sister chromatids. And there you go. Another video on DNA replication.
DNA replication begins at a specific point in the DNA molecule called the origin of replication site. Initially, the enzyme helicase unwinds and separates a portion of the DNA molecule, after which single-strand binding proteins react with and stabilize the separated single-stranded sections of the DNA molecule. The enzyme complex DNA polymerase engages the separated portion of the molecule and initiates the process of replication. DNA polymerase can only add new DNA nucleotides to a pre-existing chain of nucleotides. Therefore, replication begins as an enzyme called primase assembles an RNA primer at the origin of replication site. The RNA primer consists of a short sequence of RNA nucleotides, complementary to a small initial section of the DNA strand being prepared for replication. DNA polymerase is then able to add DNA nucleotides to the RNA primer and thus begin the process of constructing a new complementary strand of DNA. Later, the RNA primer is enzymatically removed and replaced with an appropriate sequence of DNA nucleotides. Because the 2 complementary strands of the DNA molecule are oriented in opposite directions and the DNA polymerase can only accommodate replication in one direction, 2 different mechanisms for copying the strands of DNA are employed. One strand is replicated continuously toward the unwinding, separating portion of the original DNA molecule, while the other strand is replicated discontinuously in the opposite direction with the formation of a series of short DNA segments called Okazaki fragments. Each Okazaki fragment requires a separate RNA primer. As the Okazaki fragments are synthesized, the RNA primers are enzymatically replaced with the appropriate DNA nucleotides, and the individual Okazaki fragments are then bonded together into a continuous complementary strand.
Okay, that's DNA replication in a nutshell. Now, DNA replication fits into the cell cycle in what we said was the S phase. So interphase has 3 subphases. Interphase has G1. Now, what are we doing in G1? We're making ATP, we're making proteins, we're doing normal cell functions. And then after G1, when the cell receives the appropriate signal, we enter S phase. S phase is where we do DNA replication, everything we just talked about. Then after S phase, we're devoid of ATP. We ran out of energy molecules because we just used a lot of energy to make new DNA. So what we do is we go into a gap 2 phase. By the way, the G of G1 and G2 stand for gap 1 and gap 2. So we have gap 2 phase. We're going to make ATP because we need to get ready for mitosis now. And we're going to do protein synthesis because mitosis takes a lot of proteins. It takes a lot of microtubules and other proteins. So we're going to make ATP and make proteins. And now we're ready for mitosis. Now when we enter mitosis, all we're really doing is divvying up these sister chromatids equally. But it's divided into phases: prophase, metaphase, anaphase, and telophase. Prophase is where the chromosomes condense. Matter of fact, you can't see them until they enter prophase. Metaphase is where they align in the center. We call it the equatorial line, or we call it the metaphase plate, but it's this imaginary line in the center of the cell, the equator or equatorial line. So they align in the center. Anaphase, they start being pulled apart. Now, telophase, they finish being pulled apart, plus you reform the nucleus, because the nucleus degraded before this. And you're going to reform the nucleus in both daughter cells. So you're going to actually reform 2 nuclei here. And then you do cytokinesis after that. So that's mitosis in a nutshell. Sometimes cells will leave G1 and go to what we call G0. And G0 is either a temporary or a permanent exit from cell division. For example, in humans, muscles and nerves no longer divide. I should say neurons. I shouldn't say nerves. It's actually the incorrect use of the word. Myocytes, muscle cells, and neurons no longer divide. They've entered G0. And for the rest of your life, they're not going to re-enter the cell division cycle. They're never going to divide again. But we have lots of actively dividing cells, like epithelial cells are actively dividing. Fibroblasts are actively dividing. We have some osteogenic cells in bones. These keep actively dividing your whole life. So we have lots of actively dividing cells, but muscles, myocytes, and neurons no longer do. This is a collage of all the phases of mitosis: prophase, metaphase, anaphase, and telophase. And of course, cytokinesis is pinching the cell into 2 cells. Okay, let's look at mitosis, a flick of mitosis.
This soccer player's knee will be as good as new in a few weeks, thanks to mitosis, a type of cell division that generates new cells for growth and repair. Let's move into a cell to witness the events of mitosis. Before a cell can divide, it must first duplicate the chromosomes stored in its nucleus. During chromosome duplication, several bubbles open up along the chromosome. Each bubble grows until it merges with an adjacent bubble. Each chromosome now consists of 2 identical copies called sister chromatids. Getting closer, we see that each sister chromatid consists of DNA wound around small proteins called histones. The sister chromatids begin to coil into tight helical fibers. Outside the nucleus, centrosomes that duplicated earlier move away from each other to opposite sides of the cell. Microtubules extend from the centrosomes, forming the mitotic spindle. Back in the nucleus, the DNA forms loops, becoming more compacted. These structures fold back on themselves, eventually condensing into a shorter and thicker chromosome consisting of 2 sister chromatids. As the chromosomes continue to condense, the nuclear envelope breaks up. The array of spindle microtubules is now extensive, and the chromosomes are fully condensed. Spindle fibers from each pole attach to protein structures located at the centromere of each sister chromatid. As the chromosomes are bound by spindle fibers from opposite poles, they move first one way and then another. The counteracting forces of the spindle eventually cause all the chromosomes to end up at the center of the cell, as if arranged on an imaginary plate. The sister chromatids are released from each other, each becoming a full-fledged chromosome. They are moved toward opposite poles of the cell, pulled along the spindle fibers attached to them. At the same time, overlapping spindle fibers that are not attached to chromosomes continue to lengthen, pushing the poles farther apart. Once the chromosomes arrive at their destination, they become less condensed, two new nuclear envelopes form, completing mitosis, the division of one nucleus into two genetically identical daughter nuclei. The cytoplasm divides by the process of cytokinesis, forming two separate daughter cells. In your body, millions of cells divide every second, providing new cells for growth and for repair of damaged cells.
Okay, that's mitosis in a nutshell. Another look at mitosis. It's not as probably not as detailed as the previous one.
Mitosis is a process of nuclear division by which replicated copies of a cell's DNA are organized into chromosomes. The identical copies of the DNA are then divided equally between two daughter cells. Early in mitosis, during prophase, the nuclear membrane and nucleolus begin to break down. As prophase continues, the DNA condenses to form chromosomes while the mitotic spindle, made of proteins called microtubules, begins to form and attaches to the kinetochores on each chromosome. During metaphase, the chromosomes are moved to the central plate of the cell by the spindle microtubules. In anaphase, the microtubules attached to the kinetochores of each chromosome shorten, drawing the chromatids of each chromosome to opposite ends of the cell, while the unattached microtubules elongate, stretching the cell. During telophase, the chromatids, now called chromosomes, reach the poles of the cell as the unattached microtubules continue to elongate. The nuclear membranes begin to reform as the nucleoli reappear and the chromosomes relax as the DNA unwinds. Telophase is followed by cytokinesis, or division of the cytoplasm. In animal cells, a cleavage furrow forms at the center on either side of the cell, pinching through until the original cell is divided into two daughter cells. In plant cells, cytokinesis is different due to the presence of a rigid cell wall.
Yeah, we don't care about plant cells in human anatomy and physiology, so we'll stop right there.